So we're going to go ahead and get started because we got a little bit to go over today. So we'll start by going over the homework. Okay, cool. All righty. So is everyone able to see the homework? Yeah, okay, cool. All righty, so we will start by going over the vocabulary and all the definitions. So we'll start with aneuploid. So what does it mean for an organism to be aneuploid? Right, it's going to be an individual with an error in the chromosome number. Perfect. So it's going to include deletions and it's going to include chromosome segments. Perfect. How about crossing over? What is crossing over? Anybody have an idea what crossing over is? Yes, exactly. So this is the exchange of genetic material between homologous chromosomes. Perfect. So this is going to be where you have homologous chromosomes that are going to exchange a little bit of their DNA. Perfect. So that's going to be one place where we're going to see genetic variation, okay, during meiosis. Perfect. What is a tetrad? Two duplicated homologous chromosomes. Perfect. Yeah, so it's going to be two duplicated homologous chromosomes that get bound together by the chiasma. Okay. So when you have two homologous chromosomes during meiosis, they're going to end up linking up together. Okay, and as they link up, they're going to end up forming a tetrad. Perfect. Cool. What does it mean for an organism to be euploid? What do we think? Right, Excel got that right. So this is an individual with the appropriate number of chromosomes. Okay, so how many chromosomes should a normal human have? Does anyone remember? Right, Mario got that right. So we should have 23 pairs of chromosomes. Okay, so a total of 46 chromosomes, 23 pairs of them. So if there is, if you have that number, you're perfect, you're euploid. If you have, 45 chromosomes as opposed to 46 or 47 as opposed to 46, then you would be considered aneuploid. Perfect. Wonderful. Okay, what is a reduction division? Last one. So a process by which one segment of chromosomes dissociates and reattaches to a different non-homologous chromosome? Not quite. So it's not a process where one segment of a chromosome detaches. That one's not quite it. What do we think here? First one, so it's not the first one either. The third one, right. So a reduction division is a nuclear division where you end up with half as many chromosomes as you did at the beginning. So Johanna got this exactly right. So this is where you're going to be moving from having a diploid cell to having a haploid cell, okay, at the end of some kind of division, specifically meiosis. So you end up with half as many chromosomes as you started with. Perfect. What is a synapsis? Or what is synapsis? Right, it's the pairing of two homologous chromosomes. I'm trying to find out where that is here. Where is that one at? Okay, it's somewhere here, but I'm too lazy to find it. But yeah, so it's going to be a pairing of two homologous chromosomes. So when you end up with two homologous chromosomes that end up coming together. Oh yeah, there we go. Okay, right. So when you have two homologous chromosomes coming together and actually forming a union, okay, that's going to be where we form a synapsis. Perfect. Okay. What is translocation? What do we think here? Anyone have an idea? So movement of a part of a chromosome. On the right track, yeah. So Kimberly got it. So this is the process where a chromosome dissociates and it reattaches to a different non-homologous chromosome. So for example, if we're looking at chromosome 1, for example, and chromosome 4, okay, those ones are non-homologous chromosomes. They should not be linking up with each other. Okay. But if they end up exchanging some of their genetic information across non-homologous chromosomes, that's where we end up with a translocation. So this is a type of mutation that can develop. Perfect. All right, good. How about X inactivation? Righty, so perfect. So this is where you end up with one of the X chromosomes, specifically in females, that ends up getting condensed into a single bar body that will no longer be used during that entire course of that generation of cells. Right. So this is where we end up inactivating one of the X chromosomes, specifically in females. Okay? This is not going to be a trend that happens in males because males are not going to have more than one X chromosome. Beautiful, beautiful. Okay. Next one is independent assortment. What do we think independent assortment is? Yes, Joanna. So this is where we have random assorting of our homologous chromosomes. Right. So they're going to move on regardless of whether they came from the male side or the female side, okay? Or the female, or I guess mom's X chromosomes and dad's X chromosomes. They're going to randomly assort themselves on either side of the metaphase plate. And so this is going to be the second place where we see genetic variation happening in meiosis. Beautiful. Okay, what is an autosome? Right, so it's going to be any of the non-sex chromosomes, right? So chromosomes 1 through 22, those would be the autosomes. And then the last set of chromosomes, would be the sex chromosomes, so the X and Y chromosomes. Perfect. Okay, any questions on any of these so far? Okay, all right. If not, we'll go ahead and keep going here. So next one is the proto-oncogene. Okay. So what would be a proto-oncogene? Right, so it's going to be a normal gene that's going to code for regular cell division, right? So it's going to control cell division, but if it gets mutated, then it will end up becoming an oncogene. Perfect. Okay, so this is going to help regulate the cell cycle, help kind of push the cell cycle forward. Perfect. Actually, while we're on that topic, we'll go and do the last one here. So an oncogene. Okay. So an oncogene, as we just kind of talked about, is going to be a mutated version of a proto-oncogene, right? So that's going to cause uncontrolled progression of the cell cycle. It's going to basically cause the cell cycle to push forward before it's supposed to. Perfect. Okay. What is a tumor suppressor gene? All right. It's going to be a gene that codes for a regulator protein for the cell cycle, right? So let me see where that's at here. Oh, here we go. Yeah, so it's going to be a gene that's going to code for regular proteins, regulator proteins, and that will cause a cell cycle to stop at each of those checkpoints. Okay. And that'll help to prevent against any mutations. Okay. All right, binary fission. What is binary fission? Right, Angelina. So this is the process of prokaryotic cell division, right? So prokaryotes are going to divide using binary fission and not using mitosis. Perfect. And then the last one, fertilization. What is fertilization? Anybody? Yeah, so Tim's perfect. So this is the union of two haploid cells. Okay, in this case, the haploid cells would be the sperm cells and the egg cells. Okay, so when the sperm and the egg fuse, okay, that is the process of fertilization where they're going to be bringing each of their individual sets of chromosomes together. They're going to form a diploid zygote, okay, because now you have one set of chromosomes, another set of chromosomes coming together. Now you have two sets of chromosomes within your diploid zygote. Perfect. Okay, so those are all of our definitions. We had quite a few. All right, so any questions on any of these definitions before we move forward here? No? Okay, so if not, we'll go ahead and jump forward here. So question 2, so which phase of meiosis is shown below? Is this metaphase 1, metaphase 2, prophase 1, or anaphase 1? All right, yeah, so it looks like the majority have this one right. So this is metaphase 1, right, metaphase 1. And how do we know this is metaphase 1 instead of metaphase 2? So crossover has happened. So it's we do see crossover here at this particular point. Okay. So crossover is not the key here. The attachment, yes. So the attachment of the sister chromatids. So in metaphase 1, we're going to see we're going to see our chromosomes lined up in pairs along the metaphase plate, and they're going to remain intact. Okay. Now, we were looking at phase 2. I'll just put meta 2. If we're looking at metaphase 2, okay, we would see our chromosomes lining up in a single file line along the metaphase plate. So that would be our metaphase plate. Right. So this is how we know we're looking at metaphase 1 instead of metaphase 2. Metaphase 1, you would still have your tetrads all still together. Okay. They're just lining up on either side of the metaphase plate. Okay. In metaphase 2, the chromosomes have already been separate, or have the homologous chromosomes have been separated. And so now we end up with having just these individual chromosomes lining up along the metaphase plate. All right. That's perfect, good. Perfect there, good. Okay, that's metaphase 1. Perfect, next question. So question 3, so which of the following is not true about binary fission? Okay, so we got A, it's slower than eukaryotic cell division. B, DNA replication is bidirectional. C, it occurs in prokaryotes. Or D, the daughter cells are genetically identical to the parent cell. What do we think here? Yes, perfect. So this is A, right? So binary fission, this one is false. This first one is false. So it's not slower than eukaryotic cell division. It's actually significantly faster than eukaryotic cell division. Perfect. Okay, question 4. Dora is your oncology patient. And knowing you are a cancer expert, she asks you how cancer develops. Which of the following is true about how cancer develops? So we got the cell rapidly divides with several mutations that go unchecked. We have mutations in proto-oncogenes rush a cell past its checkpoints, so necessary conditions are not met. We have mutations in tumor suppressor genes prevent the cell cycle from stopping at its checkpoints, or all of the above. What do we think? Yes, all of the above. Perfect. Yes, everyone's getting this one right. Good. So all of the above. So these are all possible reasons for how cancer can develop. Perfect. Okay, question 5. So crossing over and independent assortment occur in which stages of meiosis? So you have crossing over in prophase 1 while independent assortment is in metaphase 1. Crossover in metaphase 1 and independent assortment in telophase 1. Crossing over in prophase 2 and independent assortment in anaphase 2, or crossing over in metaphase 1 and independent assortment in prophase 1. What do we think? I see Kimberly's got this one. Yeah, so this is A. So crossing over happens in prophase 1, and then independent assortment will happen in metaphase 1. So crossing over is going to be where they're going to exchange their genetic material, and when those tetrads have linked up. Okay, metaphase 1 is where we're going to see independent assortment, and where we're going to see our chromosomes lining up on either side of the metaphase plate. Perfect. Okay, question 6. So when analyzing a group of cells, we notice that the cells spend most of their lifetime containing 2 sets of chromosomes after fertilization. Okay, which of the following best describes the life cycle of the organism that produced these cells? Are these diploid dominant, haploid dominant? Is this alternation of generations, or are we looking at binary fission? What are we thinking? Right, this would be diploid dominant. Perfect. Good job, Kimberly and Christopher. Good. So this would be diploid dominant. Okay, if we have 2 sets of chromosomes within a cell for the majority of its lifespan, it's going to be considered diploid dominant. Perfect. Now, if it only had 1 set of chromosomes after fertilization, then we would consider it haploid dominant. If it spent about equal amount of time with 1 set of chromosomes as 2 sets of chromosomes, then we would consider that alternation of generations. Okay, question 7. So what is the purpose of meiosis? Okay. We have A, to increase the genetic variation in the population, B, to form cells that are genetically identical to the parent cell, C, to act when mitosis is not possible, or D, all of the above. What is the goal of meiosis? Yes, so this one is one of those where all of the above is not correct. So the answer to this one is A. Okay, we want to increase the genetic variation within the population. Okay. So again, the goal of meiosis is to increase genetic variation and produce offspring that are unique to the parents and produce offspring that are unique to the siblings as well. Okay. So the goal here is not to produce any genetically identical daughter cells, okay, because that would be the process of mitosis. And then it's not going to be when mitosis is not possible because they're going to be used for different purposes. Okay. So the goal for meiosis is just to produce genetic variation in the population. Beautiful. Okay, question 8. Which phase of meiosis is shown below? Okay, is this telophase 2, prophase 1, metaphase 2, or telophase 1? Right, this would be telophase 2. Perfect. So telophase 2, and how do we know this is telophase 2 and not telophase 1? I'll go ahead and just kind of draw out. What would it look like if we were looking at telophase 1? Telophase 1. Anyone have an idea? How would telophase 1 look different? So you would, okay, so I'll go ahead and explain this one. So in telophase 1, this would look a little bit different because you would still see your full chromosomes. Okay. Okay. The only thing that's happened is that the chromosomes have moved to opposite ends of the cell. So remember, you had your tetrads together during metaphase 1, okay, like we saw. Let me kind of get out of this real quick. So remember in metaphase 1, okay, we had our tetrads all lined up along the metaphase plate. Okay. In anaphase 1, we pulled them apart. So you still have your full chromosomes on just getting pulled to opposite ends of the cell, but you've broken the tetrads. Okay. And then in telophase 1, you end up with individual chromosomes on either side of the, or in either side of the cell. Okay. Okay. So you still have your full chromosomes, they've just been separated onto either side of the full cell here. Okay. And then cytokinesis would happen. Okay, you would then start prophase 2 after this stage, okay, and then undergo meiosis 2 from there. Okay. Does that make sense? Yeah, okay. Okay, so that is telophase 2. So one way that you can help yourself try to orient yourself as to whether you're looking at meiosis 1 or meiosis 2 is to kind of imagine what's going on with the tetrads. Okay? If you're still dealing with tetrads and then full chromosomes, okay, then you would be looking at meiosis 1. Okay? Now, if you're no longer dealing with tetrads, where you no longer have those homologous chromosomes linked up, okay, that is going to be more likely leading you towards meiosis 2. Okay? So here we're looking at telophase 2, where we actually separated each of the sister chromatids from these chromosomes here. All right, we're gonna go ahead and move forward. So next question. So looking at Down syndrome. So Down syndrome is a disorder that is caused by a non-disjunction during sexual reproduction. Okay, so how does non-disjunction occur? So we got A, homologous chromosomes or sister chromatids fail to separate during meiosis. B, one of the X chromosomes fails to inactivate. C, a segment of the chromosome is deleted. Or D, segments of the chromosome are inverted into the genome. How does non-disjunction occur? Yeah, so Excel's got this one right. So this is going to be where our homologous chromosomes, perfect. Yeah, everyone's getting this one. So homologous chromosomes or sister chromatids do not separate properly. Okay? So if they don't separate properly, you end up having more chromosomes in some of the daughter cells than in others. And so you end up with an unequal number of chromosomes. Some cases you might have more chromosomes than you should. In other cases, you might have fewer chromosomes than you should. In either case, you end up with an aneuploidy. Perfect. Okay, number 10. So meiosis produces how many daughter cells? Is this going to be four haploid cells, 4 diploid daughter cells, 2 haploid daughter cells, or 2 diploid daughter cells? What do we think? Right, so we're going to have 4 haploid daughter cells being produced from meiosis, right? And each of those daughter cells will eventually become the sperm cell or the egg cell. Perfect. Number 11. So abnormalities in the number of X chromosomes tend to be milder than the same abnormalities in autosomes because of what? X inactivation, deletions, non-homologous recombination, or synapsis. Right, X inactivation. So if you have multiple X chromosomes, okay, all but one of them is always going to be inactivated. Okay? So if you have 2 X chromosomes, one of them is going to be inactivated. If you're born with a mutation where you have 3 X chromosomes, 2 of them are going to be inactivated. So you always have just the one being inactive, okay, or being activated. So that's going to help you to have milder abnormalities in that particular individual. Okay, number 12. So which event is taking place in the following image? Okay, so here we're looking at metaphase 1. So would we see independent assortment? Would we see crossing over? Would we see cytokinesis? Or would we see none of the above? So what event happens during this stage? So Kimberly's got this one right. So this would be where we see independent assortment, right? So crossing over is going to happen in prophase 1. Here we're looking at metaphase 1. And so metaphase 1 is where we're going to see independent assortment occur. Perfect. Okay, question 13. So P53 is a tumor suppressor gene. So how does a normal, non-mutated P53 protein work? So we got A, P53 stops the cell cycle when DNA is damaged and either encourages repair of the DNA or apoptosis of the cell. B, P53 encourages the cell cycle to continue when DNA is damaged. C, P53 encourages chromosome replication. Or D, P53 speeds up cell division by activating certain parts of the cell's genome. Yeah, so Mario and Kimberly got this one right. So this is going to be where we're going to stop the cell cycle when the DNA gets damaged. And by stopping it, that gives us time to either repair the DNA, or if the DNA is too far damaged, you're going to cause apoptosis of the cell. Okay, and that helps to make sure that you stop propagating mutations into further generations. Perfect. Okay, number 14. So 14 says, which is true about anaphase 1 and anaphase 2? So we got A, in anaphase 1, tetrads of homologous chromosomes separate, and each homologous chromosome is pulled to one pole of the cell. Okay, B, in anaphase 2, homologous chromosomes remain bound at their chiasma. C, in anaphase 1, sister chromatids separate from each chromosome, and sister chromatids get pulled to opposite poles of the cell. Or D, anaphase 2 does not operate similarly to anaphase 1 in mitosis. Which one is true? Yeah, so A is going to be true here. So the tetrads are going to get separated, right? So remember, we're having our homologous chromosome being linked up together by the chiasma, okay, in metaphase 1. Okay. When anaphase 1 happens, we're going to separate them at the chiasma and pull each individual homologous chromosome to opposite ends of the cell. Perfect. Okay, last question. During meiosis, when does the ploidy, the number of sets of chromosomes, get reduced from diploid to haploid? Okay, is this going to happen when telophase 1 is complete, when prophase 1 is complete, when telophase 2 is complete, or when anaphase 1 is complete? Okay. When do we have that reduction division take place? Yeah, so it's going to happen after telophase 1. Okay. So remember, in telophase 1, we have two sets, sorry, at the beginning of meiosis 1, and we're going to have two sets of chromosomes. Okay. When telophase 1 occurs, okay, we end up undergoing cytokinesis, and then we end up separating each of those sets of chromosomes into their individual cells. Okay. And then at that point, you end up having just one set of chromosomes. So you end up having a haploid cell. Perfect. Okay, that is that. Sorry, it's a bit of a long homework assignment. Are there any questions on this homework before we move forward here? Okay, so if not, there is one thing I do want to show you guys for this week. So this week we'll be talking about genetics. So we did this lab before spring break started. So today, or this week, we're going to be looking at genetics in a little bit more detail. And I want to show you an extra credit assignment that is going to be due next week, or 2 extra credit assignments that are going to be due next week. Okay? So if you go over to chapter 8, patterns of inheritance, and that's going to be our genetics chapter for this week, you're going to see the 2 PowerPoints, and then at the bottom of those, you're going to see some extra credit assignments. Okay? So one of them has to do with simple dominance and simple recessiveness, which we'll be talking about today. Okay. The other one is going to be on non-Mendelian genetics, which we'll be talking about on Thursday. Okay. So each one of these is worth 10 points. So you get a total of up to 20 points extra credit, okay, depending on how you do. This will be due next Tuesday by midnight. Okay. So you have all week to knock this out. You have the weekend. And again, like I said, this will be an extra credit, up to 20 points extra credit for you to earn back. Okay, so there's a bunch of problems on simple dominance and simple recessiveness and how to do these Punnett squares and everything. You'll turn that in next week. I'll have a place for you to turn that in, and then you can earn your points there. Okay, so I'll talk more about that at the end of today. But for now, what I want to do is we're going to go ahead and dive into our chapter for this week. So, is everyone able to see the PowerPoint now? Yeah, okay, cool. Okay, so today we're going to be looking at genetics in a little bit more detail than we looked at it in lab before we had spring break. So this first part is going to be more history. I'll tell you when to start taking notes. For now, just listen, and we'll kind of just play with our experiment, or play with Mendel's experiments. But I'll let you know when to start taking notes. So just kind of bear with me for now. There's a little bit of history here that you don't need to worry about. Okay. But before we get into that, quick question. So what is the purpose of meiosis? Is it A, to produce offspring that are genetically identical to their parents, or B, to produce offspring that are genetically unique? What do we think here? Right, we want to be producing offspring, beautiful. So everyone's getting this one right. So we want to be producing offspring that are genetically unique, right? We want them to be unique to the parents. So we want them to be genetically different from the parents. And we also want them to be genetically different from their siblings as well. Okay. So today what we're going to be taking a look at is how it's possible for siblings to end up looking a little bit different. Last week we looked at how you can end up with genetic variation through crossing over, as well as through independent assortment. And through these different processes, we can end up having all kinds of different types of genetic variation in the sperm cells and the egg cells that we end up producing. Okay. So today we're going to be looking at what happens when we unite those sperm and the sperm cells and egg cells. Okay, when fertilization happens, what's the chances of us producing an offspring with a certain trait? Okay, so we're going to be taking a look at how that's able to happen. Excuse me. Okay, so again, for now, don't worry about taking notes. I'll let you know when to start taking notes. Okay. So there was this guy in the mid 1800s. His name was Gregor Mendel. And so he was a teacher, he was a scientist, and what he wanted to do was try to study how people inherit traits from their parents. Okay. And so he was looking specifically at honeybees and looking at plants to see how different traits would be passed on from generation to generation. Okay. So in about 1856, he decided that he was going to start using some pea plants, okay, as a model for studying inheritance. They were going to be his model system. Okay. And as we'll see, the data that he came up with, the results that he came up with, ended up becoming the foundation for our current understanding of genetics. So we consider Gregor Mendel to be the father of modern genetics here. Okay. So at the time Mendel was studying all of this stuff, most other scientists believed that whenever traits were inherited from their parents, okay, or an offspring's traits were inherited from parents, those traits were a blend of the parents' traits. Okay, so for example, if you were looking at, for example, a parent who had, brown hair and a parent who had blonde hair, okay, their offspring would have something in between, a combination of brown and blonde hair. If you were looking at offspring that had a tall parent and a short parent, the offspring would be a medium parent or a medium child. Okay. So they believe that all traits across the board, okay, were always going to be inherited as a blend of the parents' traits. Okay, so it's going to be an intermediate. So this is what they called continuous variation, where you see a range of small differences in between kind of two extremes. Okay, that's called continuous variation. Okay. Now Mendel, he decided to kind of go away from that. He wanted to work with traits that have that showed discontinuous variation. And a lot of scientists at the time didn't really think discontinuous variation was a thing. And so what discontinuous variation is where an individual inherits one of two traits. Okay, so for example, if a parent has black hair and another parent has brown hair, the offspring will either inherit black hair or brown hair, not a combination of the two, but you'll inherit one or the other. Okay? So offspring will inherit one of two easily distinguishable traits. Okay? That's what discontinuous variation was. Okay? Now, like I said, a lot of scientists at the time didn't really subscribe to this idea. They all thought that it was all continuous variation. Offspring were always a blend of all their traits between both of these two parents. Okay? But Mendel didn't believe so. So he started looking at discontinuous variation. Okay? And so when he started to study this, he actually started to see a few results. Okay? And so the results showed him that, in fact, different traits from offspring, okay, are not always going to be a blend of the two parents, okay, of the two parental traits. Okay? Some traits are going to be inherited as distinct traits. You either inherit one or the other for some traits, while some other traits can also be inherited as a blend. Okay? But he found that some traits can be inherited distinctly, either one or the other. Okay? Everyone kind of following me so far? Okay. So one quick example was when you were looking at some violet flowers and white flowers. Okay? If you kind of allow those two to mate with one another, okay, the offspring would either be violet or white. Those are some of the results that he would see, and we'll look at that going forward too. Okay. So the pea plant that he decided to use was called the garden pea, or Pisum sabatum, and so this had three different advantages to it. Okay, the advantage to using this particular garden pea, number one, is that it self-fertilizes, meaning that it's able to fertilize itself. It's able to produce offspring, okay, from its own self. Okay? And so this would allow you to be able to produce these inbred, true breeding plants and produce offspring that look exactly the same as parents. Okay? That's number one. Okay? Number two is that the garden pea is able to grow very, very quickly. Okay? By the time it reaches maturity, that can happen over the course of a single season. Okay? And so you can actually grow multiple generations of these garden peas over a very short period of time, and you can actually evaluate them again over a very short period of time because they grow so quickly. Okay? And then the last advantage to using the garden pea is that because they grow to maturity so quickly, you can produce large amounts of garden peas at once. Okay? and grow them all simultaneously. And that would give him a very large sample size to study when he was looking at his results. Okay? And so that was able to show him that his results were not happening just randomly or happening by chance, and that his results were actually being caused by some actual mechanism. Okay? So those were the advantages of the garden pea. Okay? So what he would do was he would take, in order to kind of form a hybrid, okay, he would take two true breeding plants, two true breeding individual plants, and he would take the pollen, which contains the sperm for one plant, and add it to the stigma of another plant, which had the eggs. Okay? So when he took the pollen and added it to the stigma of another plant, that would help him to form some seeds, and the seeds that would result from that would end up forming the hybrid plant, okay, the offspring between A and B here. Okay, so that's how he would cross these plants. Okay, so this is the part where I want you to take notes at. Okay. So when he was looking at his generations here, okay, the first generation was called the P generation, or the parent generation. Okay, these were the plants that he was starting with. Okay. So what he would do is he would cross these two plants and or crossing is another way of saying they would allow them to mate. Okay, so he would mate these pea plants with violet flowers, okay, with these pea plants with white flowers, okay, and these two made up the parent generation. Okay. Now, when they mated with one another, they would produce some seeds. Okay, those seeds will be planted, and then the seeds, or the plants that would sprout up from those seeds, would end up becoming the plants in the F1 generation, or the filial generation. Okay, so the F1 generation would result from the two parents. Okay. Then at this point, he would then analyze the results that he would see in the F1 generation, and then he would allow the F1 plants to then self-fertilize with themselves. Okay, so they would take the pollen, add it to its own stigma, okay, and then allow them to fertilize with itself. Okay. Once it fertilizes with itself, and it's going to form some more seeds, you take those seeds, and then you plant those seeds. Okay, and the plants that sprout up from those seeds end up becoming the F2 generation. Okay, so these would become the offspring of the F1 generation. And so he would do this for several generations, but the ones that were key were the P generation, the F1 generation, and the F2 generation. Okay, those were the key for what Mendel was actually studying here. Okay. Okay, so when Mendel was looking at his results, he not only looked at just the color of the flowers, but he actually looked at 6 other traits. So a total of 7 distinct traits. So he would look at plant height, whether they were tall or short. Okay. He would look at the flower color, whether either violet flowers or white flowers. He would look at pea pod color, which was either green or yellow, and then a bunch of other different characteristics as well. Okay. Okay, but they were looking at specifically each characteristic with 2 individual traits, either being tall or short, violet or white colors for the flowers, or green or yellow pea pod colors, or a number of other possible characteristics. Okay. And so again, when we're thinking about a trait is just a physical appearance of a particular characteristic that's heritable, okay, a characteristic that you can inherit. Okay. And so in doing so, he generated thousands of F2 plants, okay, over a very short period of time. Okay. So when Mendel was doing all these experiments, he started to see some very distinct results. Okay, so this is going to be kind of key here. So when we're looking at flower color of these pea plants, okay, he made sure that everything about those pea plants was exactly the same, except for the flower color. Okay, everything else was identical. Okay, so we had some pea plants that had purple flowers and some pea plants that had white flowers. Okay, these would make up our P generation, our parent generation. Okay, so they either had one or the other. Now, when these 2 plants were able to cross with one another, okay, what he found was that all of the offspring, okay, 100% of the offspring in the F1 generation, all had purple flowers. Okay? And so in this generation, white flowers completely disappeared from the F1 generation. Okay. Everyone kind of follow with me so far? Okay. Okay, so going back to what the other scientists believed at the time. So during Mendel's time, most other scientists believed that continuous variation was the actual design of inheritance patterns. So what would they have assumed would happen when crossing plants with white flowers with plants with purple flowers? So we got A, they would assume that all plants would have purple flowers. B, they would assume that all plants would have white flowers. C, they would assume that there would be a random assortment of plants with purple and white flowers. Or D, they would assume that all plants would blend the purple and white flowers, or white colors in the flowers, to produce plants with light purple flowers. What would other scientists have believed at the time? Or what would they have believed should have happened? Yeah, so Jason and, yeah, so everyone's getting this one right, perfect. So other scientists would have assumed, okay, that you would have ended up with a blend of the two colors. It would be a combination of both purple and white, so you would have end up with light purple flowers. Okay, but in fact, that's not what we saw. Okay. What we ended up seeing was that all of these plants in the F1 generation ended up being fully purple. Okay. Okay. So they all ended up fully purple here. Now, once Mendel found his F1 generation, the next thing that he would do is he would allow the F1 plants to self-fertilize, okay? Allow them to fertilize with themselves. Okay? And so when he allowed each individual F1 plant to self-fertilize, we would then see in the F2 generation, 3 plants that would be purple, and then 1 plant that would be white. Okay, we'd see a 3-to-1 ratio of purple and white flowers. Okay. And so what we see here is that the white flowers began to reappear in the F2 generation. Okay, so we saw white flowers appear in the parent generation. It skipped a generation here in the F1 generation, and then it reappeared here in the F2 generation, which is kind of curious. Like, why would the white plants start to show, or why would the white flowers show up in one generation, but not in the previous generation? Okay, so, and on the flip side, we would see purple flowers show up in all of these generations here. Okay. So this is kind of a curious kind of results here. Okay. But keep in mind here, in the F2 generation, he always saw a 3-to-1 ratio. Okay. A 3-to-1 ratio of, one trait to the other. Okay. And so in order to kind of make sure that his data wasn't being kind of affected by anything, what he did was a reciprocal cross. So basically what he did was he would go back to the parent generation. Okay, and instead of taking, for example, the pollen from the purple flower and adding it to the white flower, he would take the pollen from the white flower and add it to the purple flower. Okay. So he's doing the same thing, but doing everything in the opposite order. Okay. Instead of taking pollen from the purple flower, he would take it from the white flower and add it to the opposite flower. Okay. And what he saw was that the ratios did not change at all. He would still end up with the exact same results, okay, regardless of where the pollen came from. Okay. Again, remember that pollen contains the sperm. So saw the same ratios, same numbers, same results all the way across the board, regardless of where that pollen came from. So then when he started looking at the other characteristics, he saw that those same other characteristics worked exactly the same way. Okay. When he was looking at plants as far as their height, okay, we saw a tall plant and then a short plant, allow them to cross with one another. In the F1 generation, they all end up being tall. Okay. When you allow them to self-fertilize, then you end up with seeing a 3-to-1 ratio of tall plants to short plants. Okay. Same exact numbers, same exact ratios, exactly the same as we saw with the flower. Okay. Same thing with the pea seed colors. Okay. Here we see yellow seeds and we see green seeds. In the F1 generation, they all end up being yellow. Okay. And then in the F2 generation, we ended up seeing the same 3-to-1 ratio, 3 yellow plants or yellow seeds to 1 green seed. So all these characteristics, they work exactly the same, regardless of the trait he was looking at. Okay. So based off of Mendel's results, he started to be able to make a few conclusions. Okay. One of the conclusions that he came up with was that there were some traits that were going to be considered dominant, meaning that they were always going to be expressed, or there were going to be some that would be considered recessive. Okay. So dominant traits are traits that will always show up from one generation to the next. Okay. They will always show up in every single generation as long as that trait is present. Okay. Recessive traits, on the other hand, have the ability to disappear from one generation entirely and then reappear in the next generation. Okay. So those were one particular set of traits that were incredibly important, or one particular conclusion that was really important for what Mendel was able to find during his experiments here. Okay. So dominant traits were always inherited unchanged, okay, while recessive traits were able to disappear and reappear from generation to generation. All right. Whoops. Okay. Okay, so question here. So which of the following correctly describes the inheritance pattern of the flowers in Mendel's experiments? Okay, so we got A, the purple flower was a dominant trait and the white flower was recessive. B, the white flower was dominant while the purple flower was recessive. C, both purple and white flowers were dominant. Or D, both purple and white flowers were recessive. Yeah, so Jason and Mario got this one right. So this is where we see the purple flowers being a dominant trait because we see that being expressed in every single generation. Okay? While the white flower is recessive because we see it in the parent generation, we don't see it in the F1 generation, but then it reappears here in the F2 generation. Right. Okay. Any questions so far on what Mendel was doing here? Okay. Okay. So by being able to determine that some traits were dominant and other ones were recessive, he was able to make a whole set of other conclusions that would eventually become the foundation for our modern day understanding of genetics. Okay. So being able to see that there were two distinct flower colors, okay, and seeing that some traits could reappear and disappear from generations, helped him to see that there were traits that were separate from each other. Okay, like we were seeing purple flowers and white flowers, okay, that were completely separate, okay, but they do not blend with each other. Okay, so there's some traits that we know are not going to blend and that they're going to be inherited separately. Okay, one or the other will be inherited. Okay, so what that tells them is that each individual has to have two copies of a particular trait. Okay, two copies of that trait are going to be found in each organism. Okay, and so each of those parents is going to contribute one copy of that trait. Okay, one parent is going to contribute one copy of that trait. The other parent is going to contribute another copy. So the offspring ends up with two copies. Okay, and that's going to be the case for any trait that we look at. Okay. Now, based off of that, he was then able to determine, okay, that there were some traits that were going to be dominant traits. Okay, so if a dominant trait was showing up in a particular organism, what that meant was that there had to be either two copies of the dominant trait for us to see the dominant appearance, or there had to be one copy of the dominant trait and one copy of the recessive trait, and we would still see that dominant appearance, okay, the dominant physical appearance. Okay. The counterpart to that is that in order for us to see a recessive appearance or a recessive trait, an organism has to have two copies of the recessive trait only. Okay? There cannot be any dominant traits for us to be able to see a recessive appearance or recessive characteristic. Okay. Okay. So all of these conclusions, like I said, end up kind of forming the foundation for our modern day understanding of genetics here. Okay, so that's going to bring us to the laws of inheritance. Okay, so one thing I do want to bring up kind of more historically is Mendel kind of finished doing, he started this experiment with the pea plants in about 1856, and he finished it at around 1865. Okay, he stopped it around 1865 because he ended up joining a church, became a pastor, and then just basically left science behind. Okay, now during that time, maybe it was around 1868 or so, but late 1860s. During that time, okay, no one really paid attention to Mendel's work at all, even though he ended up being right. And so people kind of disregarded his work until about 1900. So over 30 years later is when people started to rediscover Mendel's work and then start to think, okay, maybe this Mendel guy was actually onto something. And so his work was kind of rediscovered. It was started to be able to see that, okay, this actually is correct. And now we can actually look at some laws of inheritance that are actually based off of what Mendel was able to determine. Okay? And so that is going to bring us to this next section, which is where we're able to apply Mendel's work, okay, into our current understanding of genetics. Okay? And so when we're looking at genetics here, genetics is the study of how genes are going to be inherited, okay, from one generation to the next. Okay, so remember that when we're looking at chromosomes, we're going to have two pairs of chromosomes, okay, or a pair of chromosomes, okay, for each individual set. Okay, 23 pairs of chromosomes. Each one is going to be found in twos. Okay, so these would be considered homologous chromosomes. Okay. In homologous chromosomes, they're going to have the same set of genes. Okay. So remember that a gene is just a region on a chromosome that is going to code for some characteristic. Okay. In this case, we're looking at eye color here. Okay. So each of these is a homologous chromosome. Here we have a gene for eye color. Here's another gene for eye color. Okay. So individuals are going to have two copies of each chromosome. Okay. Again, one that came from mom and then one that came from dad. Okay. And so each of those is going to come from the gametes, coming from the sperm, which will be carrying one of those copies, and then the egg, which will be carrying the other copy. Okay. So we're going to have two of each chromosome, and as a result, you're going to end up having two copies of each individual gene, okay, just because you have two copies of the same chromosome. Okay. And so when our chromosomes get separated during meiosis, okay, the sperm will contain one set of chromosomes, the eggs will contain another set of chromosomes. When they join together during fertilization, that is going to be where we join those sets of chromosomes, and you end up with a diploid zygote, okay? Having two sets of chromosomes, and as a result, two copies of the same gene. Okay. Okay. Now, the genes that we inherit from homologous chromosomes are going to come in different variations. Okay. So, for example, if we're looking at eye color, we can have people who have brown eyes, we have people who have blue eyes, we have people who have black eyes, we have people who have green eyes, and so on and so forth. So those are all different variations of the same gene. Okay. All of those different gene variants are what we refer to as alleles. Okay. So alleles are gene variants for a particular homologous chromosome, and they're going to be found in the same, in the same region, in the same gene. Okay. So for example, here we're looking at purple flowers and white flowers. So here we would have an allele for purple flowers on one chromosome, okay, which was inherited from one parent, okay, and on the other chromosome, inherited from the other parent, we might have an allele for white flowers. Okay. So these are variations of the same gene, okay, but their DNA sequences are a little bit different. Okay, yeah. So these are like Paul said, this will be get posted later. So these are all variations of different genes. So genes are always going to be found in the same spot, okay, but the DNA sequences within that particular gene are going to be different. Okay. Those different gene sequences are going to be what we refer to as alleles. Okay. And so we're going to signify our alleles as either being dominant alleles or recessive alleles. Okay. So dominant alleles are always going to be signified by a capital letter, like we see with this capital P here, while recessive alleles are always going to be signified by a lowercase p. Or a lowercase letter. Okay. Now, depending on the alleles that you inherit from your parents, you may or may not inherit two copies of the same allele. Okay. So, for example, if you have two parents that both have brown hair, for example, then you're going to inherit two copies of the brown hair allele. Okay, one from mom and one from dad if they both have brown hair. Okay. However, it is possible to inherit two copies of different genes. So you can inherit two different alleles. If you have a parent who has brown hair and one parent who has blonde hair, okay, you might inherit one copy of the brown hair allele and one copy of the blonde hair allele, kind of similar to the purple flowers and white flowers here. Okay. Here we have an allele with the purple flowers and an allele with white flowers. Okay. So you may or may not inherit two copies of the same allele. It just depends on which allele you inherit from your parents. Okay. But again, one thing to keep in mind is that in a population, you're going to have more than two alleles in a population. Okay. For example, you might have more brown hair, blonde hair, brown hair, blonde hair, black hair, red hair, and so on and so forth. Okay. So more than two alleles are going to exist in a particular population. Okay, so that being said, that's going to lead us into our phenotypes and genotypes. Okay, so our alleles that we end up inheriting from our parents, okay, are going to interact with one another. Okay, and as they interact, they're going to form our phenotype and they're going to form our genotype. Okay? The phenotype is the observable physical trait that we're able to express. Okay, that's going to be what we actually are able to see on each other. Okay, our phenotype is our physical appearance. Okay, the genotype is going to be the genetic makeup of the organism. Okay, and so the genetic makeup is going to be what's found specifically in your DNA, within your chromosomes here. Okay? And so one thing to keep in mind here is that the genotype will always determine the phenotype, which makes sense because your DNA sequences, your DNA is going to determine your physical appearance. It's going to determine what you look like and how you appear. Okay? So genotype will always determine your phenotype. Okay, so here's just a couple of examples. Okay, so phenotype is the observable trait. So here, if we're looking at our pea seeds, here we have some seeds that are yellow and some seeds that are green. Okay, so the color of these seeds would be the phenotype. We have one yellow, one green. That would be the physical appearance of those seeds. So that would be showing us our phenotype there. Okay. The genotype is where things get a little bit more complicated. Okay. So the genotype is going to be determined by the pair of alleles that you inherit. Okay? So again, remember, you're going to have two copies of alleles within our chromosomes, one on each homologous chromosome. Okay? So for example, here we have a big Y allele and another big Y allele. Okay? So the genotype would be big Y, big Y. Okay? Here we have one little Y allele and then another little Y allele. So the genotype would be little y, little y. Okay? So the genotype is the combination of the two alleles in that particular trait. Okay? And then in this case here, we have a big Y allele and a little y allele. So the genotype would be big Y, little y. Okay? So we're going to be classifying these genotypes in a very specific manner. Okay? If you have two copies of the same allele, like we see with big Y, or little y, okay, we consider that to be a homozygous individual, okay, or homozygous for that trait. Okay. If you have one copy of each allele, like here we have a big Y allele and a little y allele, okay, then we refer to that as a heterozygous trait or heterozygous individual. Okay. So what we see in this F1 generation is we see all of our seeds being yellow. Okay. So 100% of our seeds end up becoming yellow in this generation. Okay. And then if we look at the F2 generation, we see yellow seeds, and then we also see that the green seeds have reappeared in this generation. Okay. So keep that in mind. We'll kind of play with that a little bit more as we go forward here. Okay. But if we look at the F1 generation, we see that the F1 plants are different genotypically from the two parent plants. Okay. We have big Y, little y, and then in the F1 generation, we have big Y, little y. So the genotype is different from both parents here. Okay? So we're able to form genetically unique individuals, okay, in the F1 generation. Okay. Okay. Now, when we're looking at each of these genotypes, we're going to be classifying them in a little bit more specifically here. Okay, so if we look at homozygous individuals, okay, we need to have a way of being able to be more specific with that term. Okay. So like I said, homozygous just means you have two copies of the same allele, either big Y, or little y. However, to be more specific, we're going to be classifying them as either homozygous dominant, if they have two copies of the dominant allele, or homozygous recessive, if they have two copies of the recessive allele. Okay. So you have two possible ways of being homozygous, but to be more specific, you either classify it as homozygous dominant or homozygous recessive. Okay. Now, when we're looking at heterozygous individuals have one copy of each allele. So they're going to have two different alleles for that particular characteristic. Okay? So they're going to have a dominant allele and then one recessive allele. Okay? So we call these guys heterozygous. Okay? There's no reason to call them heterozygous dominant because it's understood that they have one copy of the dominant allele if they're heterozygous. They're either dominant or recessive. So here, heterozygous are always going to have one copy of the dominant allele, so we just call it heterozygous and just leave it at that. Okay. So Like we see here, they're going to have one copy of the dominant yellow seed allele, okay, and then one copy of the the recessive green seed allele. Okay. Now, that being said, looking at the F1 generation, we see that all of our offspring here, 100% of our offspring, show up as having yellow seeds, even though they have one copy of the dominant yellow seed allele and one copy of the green seed allele. Why would that be the case? Then we have an idea why we would see all yellow seeds here instead of seeing some green in the F1 generation. Right, yellow is dominant, exactly. So the yellow is dominant, and so it's going to be showing up and masking the other one. So we'll take a look at that in a little more detail going forward here. Okay. So this is going to lead us into the law of dominance. Okay. So before we get into the law of dominance itself, there's going to be two characteristics I want us to kind of be paying attention to. Okay. When we're looking at homozygous dominant individuals here, okay, so we have a big Y allele. That's going to give us our yellow phenotype. Okay. And then our heterozygous individuals, which will also show up as yellow, okay, they're going to look identical. So looking at a homozygous dominant individual and a heterozygous individual, they're going to be identical. Okay. They're going to have the same physical appearance for those particular traits. Okay. Now, recessive traits or recessive organisms, homozygous recessive organisms, are only going to show their recessive traits if they only have their recessive alleles available. Okay. So recessive alleles are only going to show up in homozygous recessive organisms. So here we have the homozygous recessive, little y. Those are going to be the green seed alleles here, the green seed organisms here. Okay. And they're going to only show up whenever you have a homozygous recessive organism. Okay. You will never see a recessive trait when it's also paired up with a dominant trait. Okay, it just won't happen because dominant will always mask it. Okay. So that's going to lead us into the law of dominance. Okay. The law of dominance tells us that in a heterozygote, okay, the dominant allele will always completely mask the recessive allele. Okay. So here we see our homozygote. Here we have our big Y, little y. Okay. The dominant allele, the yellow allele, is always going to mask the recessive one, the green one. Okay. So therefore, only the dominant allele is only going to be seen. Okay. That's going to be the only effects that will be seen in a heterozygote. Okay. And so that's what we see here with these pea seeds here. If we also look at the flowers, we're also going to see the same kind of general trend. Okay. Here we have purple flowers in our pea plants. Here we have white flowers. Okay. In the F1 generation, we're going to form a heterozygotes, but they're always going to be purple. Okay. And that's going to be because one trait was dominant, the other one was recessive, and so the dominant trait masked the recessive white flower trait here. Okay. So in heterozygotes, the dominant allele will always mask the recessive allele. Okay. The only way that you're going to see a recessive trait is if you have two copies of the recessive allele. Okay, so if you're homozygous recessive, that's the only way you'll ever see a recessive trait. Okay. Any questions so far? If this makes sense, go ahead and throw a check mark or a heart or something, so I kind of know where everyone's at here. Make sense? Okay. Okay. All right, so that being said, that we talked about. So heterozygotes are going to have one copy of the dominant allele and one copy of the recessive allele. Okay, so however, that recessive allele, even though it's not being expressed, is still found in their DNA. Okay, it's still found in their chromosomes. Okay, and as a result, that recessive allele, even though it's not being expressed, can still be passed down to their offspring. Okay, so we have an example here of albinism. Okay, so albinism is a trait that is a condition that is homozygous recessive. It's a recessive genetic disorder. Okay, like I said, in order to get a recessive trait, you have to have two copies of the recessive allele. Okay, so I'll just put little a here. Okay, that's going to give us albinism. That's going to be this guy over here. Okay, this little boy. Okay, so in order for us to get a homozygous recessive individual, they have to inherit one copy of the recessive allele from both parents. Okay? So here's this little boy's mother, okay? And this little boy's mother has normal skin pigmentation. Okay? So she's producing normal skin pigmentation, but she had an offspring. She had a little boy here, okay, who has albinism. Okay, he's not producing any skin pigmentation. Okay, so we're going to kind of draw out what we would think her phenotype would be. And so her phenotype would be something like big A, little a. Okay, she has big A because she's still expressing the dominant normal skin pigmentation trait, but she had to have an offspring, okay, that had the little a allele because it's recessive. So she ended up passing down one of those recessive alleles. Okay. Now, if we look at the little boy's dad, let's assume he's also big A, little a. He's also a heterozygote. Okay. Okay. So he may also have normal skin pigmentation, but if he also happens to pass down this recessive allele, now all of a sudden, their offspring now has 2 copies of that recessive trait. Okay. And so as a result, the little boy will end up developing albinism. He ends up being an albino child here, okay, with no normal skin pigmentation. Okay. So even though the 2 parents themselves have normal skin pigmentation, they still express the dominant trait, it is still possible for them to pass on the recessive allele, okay, if that's part of their genotype. Okay. And so, like I said, yeah, each one of those alleles is going to be inherited from each parent here. Okay. Does this make sense? Okay. So again, the key here is that in order to see a recessive trait, you have to inherit 2 copies of the recessive allele, one from mom and one from dad. Okay. But even though the parents may themselves be expressing the dominant trait, it is still possible for them to pass on the recessive allele. Okay, so that is how recessiveness works here. Any questions going forward here? Okay. Okay, cool. So with that, that's going to get us to a point where we're going to try to identify the probability of having offspring with specific traits. Okay. So one thing that we can do is called a monohybrid cross. So a monohybrid cross is where we have fertilization of 2 true breeding parents. True breeding is another word for saying homozygous or homozygous parents, either homozygous dominant or homozygous recessive. Okay. But they're only going to differ in that one particular trait. Okay. The offspring that result as a result of monohybrid cross are what we would end up calling monohybrids. Okay. So monohybrids are the offspring of 2 true breeding homozygous parents. Okay. Now what we see is that in the F1 generation, all of the phenotype or all of the offspring have the same phenotype as one of the 2 parents. Okay. And then in the F2 generation, we see a 3-to-1 phenotypic ratio, okay, where the offspring have a 3-to-1 ratio of one particular trait and then one having the other. Okay. And then again, the goal here is that, or the key here is that each parent is going to contribute one of its 2 alleles to its offspring. So that brings us to this last, or kind of this kind of big picture part of our chapter here. We want to be looking at Punnett squares. Okay. So as geneticists, one of the things that we want to be able to do is we want to be able to predict the phenotype and the genotype of an offspring based on the parents' genotypes. Okay. We want to be able to determine, the different traits that an offspring might develop based off of the parents' DNA. Okay. And so we're going to be using some Punnett squares to help us be able to identify those particular traits here. Okay. So these are going to be some tools that we're going to be using. And so we're going to kind of break down how to use a Punnett square. Okay. So we've seen this in lab already, so I'm not going to kind of beat this over you guys' head too much. But when we're doing a Punnett square, what we're going to do is we're going to draw a grid kind of in a 2x2 grid here. And what we're going to do is we're going to list each parent's genotype, okay, along the 2 sides of, or along the top side and the left side of the Punnett square. Okay. So one parent's genotype is going to be across the top, so that's going to be big A, big A. That's going to be, we'll just call this dad. Okay. I'll put dad over here. Okay. And then we'll have little a as the genotype for mom on this side. Okay. Now, each of those genotypes, oops, sorry. Each of those genotypes is going to be listed across the side. Okay? And so each letter of these genotypes is going to refer to one of these alleles. Okay? And each of those alleles will be found in the gametes. Okay? So for example, this might be a sperm cell that contains the big A allele. Okay? This would be another sperm cell that contains a big A allele. Okay, for dad. Okay, on mom's side here we have an egg that contains a little A allele, and then another one over here that contains a little A allele. Okay. So then at this point, the next thing that we're going to do is we're going to kind of simulate what we think would happen if each of these sperm cells and egg cells fertilize each other. Okay, if fertilization took place. Okay, so if this sperm cell joins up with this egg cell, what's going to be the genotype of the resulting zygote? Okay. So here we have a big A, little a. So we're going to have big A, little a in this particular square over here. Okay. This square, we're going to have big A, little a, big A, little a over here. This one, we're going to go from this side over here to this side over here. We're going to have big A, little a on this side, and then big A, little a in this square over here. Okay. So these are going to be the genotypes of the zygotes that we produce, okay, or of our offspring. Okay. Okay. So that's how we're going to organize our Punnett square. Now, we have a question here. So in human populations, brown eyes are a dominant trait while blue eyes are recessive. Okay, so Tony Stark and Pepper Potts are having a baby. Tony is homozygous with brown eyes, so he's big B, big B. And Pepper also has brown eyes, but she is heterozygous. So what is Pepper's genotype for eye color? What do we think? Is it big B, big B, big B, little b, or little b, little b? What is Pepper's genotype? Right, so her genotype is going to be big B, little b. Perfect. Right, big B, little b is going to be her genotype because she is heterozygous. Perfect. So now we're going to try doing the Punnett square. So we'll go ahead and do this Punnett square this way. And hopefully I can draw out my squares a little bit more straight than I did last time. Okay. So if we know that Tony, we'll put Tony in blue. Tony is homozygous dominant. We're going to list his genotype across the top. And it doesn't matter which side you do it as long as you do it correctly. So he's big B, big B. Okay. While Pepper, on the other hand, so I'll just put P for Pepper over here. Okay. Pepper's genotype was big B, little b. Okay. So I'll list her genotype over here. And big B, little b. Okay. And so now we're going to go ahead and do our Punnett square. Okay. So in our top left box, what genotype should we end up with in this case here? Right, big B, big B. Perfect. Big B, big B. How about in the top right box? Big B, big B. Perfect. And then in the bottom left, big B, little b. Perfect. And then bottom right, big B, little b. Perfect. Okay. So now, one thing that we need to do, not just after we do our Punnett squares, we need to be able to analyze our Punnett square and be able to determine what it means. Okay. So we're going to kind of look at this in a couple different respects here. So if we're taking a look at the probability, let's just say probability of a homozygous dominant offspring. What's the probability of having offspring that is going to be homozygous dominant? 50%, right. 50% because we see 2 squares of our 4 having the homozygous dominant genotype. Oops. Okay, perfect. Now what about the probability of of heterozygous? Right, 50%, because again, 2 of our 4 boxes have the have the heterozygous genotype. Okay. Now next one, so we looked at it in terms of the genotype now. what's the probability of having offspring that have brown hair? Or what was it? I think brown eyes, which was the dominant trait. 100%, right, because all 4 of our boxes contain at least one of the dominant alleles. Okay? And so kind of extrapolating from that, the probability of having blue eyes would be 0%. And because again, the only way we're going to see blue eyes is if we have a homozygous recessive individual. Okay. So when you're doing these Punnett squares, you need to be able to determine what's going on both genotypically and the probability of each individual genotype and the probability of each of the phenotypes. Okay. Okay. Any questions on how to do the Punnett squares here? Okay, cool. Okay, so now we have another example, and I think this will probably be the last example we do before we cut ourselves short today. Here we're looking at some offspring, or some parents, our parent genotype. Okay, so here we have big Y, and little y, little y. Big Y being our yellow seeds, little y being our green seeds. So if we allow these two to mate with one another, okay, we're going to go ahead and do our Punnett square again. So if we do our Punnett square here, we're going to list our genotypes along the side. Okay, so we're going to have big Y up here from one parent, and then little y for the other parent. Okay, and so now we're going to go ahead and try our Punnett square. So what should we end up with in this square here, in our top left? Right, we would have big Y, little y. Okay, what about in the top right? All right, we see big Y, little y, perfect. Okay, bottom left, big Y, little y, perfect. And then bottom right would be big Y, little y, perfect. Okay. So now if we try to analyze our Punnett square based off of what we've done here, okay, what is the probability of a homozygous dominant offspring? Right, 0%. Okay. What about the probability of homozygous recessive? I'll just put homozygous recessive. What do we think? Right, 0%, because again, we don't see 2 copies of our recessive alleles here. Okay, and then what about heterozygous? What's the probability of having a heterozygous offspring? Right, 100%, because all 4 of our 4 boxes have the heterozygous genotype. Okay. So now that being said, what's the probability of having yellow offspring? What do we think here? Right, 100%. 100% because all 4 of our boxes contain at least one of the dominant alleles. Okay, remember that dominant is going to be the yellow allele here. Okay. And then as a result, probability of having green would be what? 0%, perfect. Right, so when you're going through your Punnett square, you need to be able to understand and analyze your Punnett square, both based off of their genotype and then again based off of their phenotype here. Okay. So this is what we would see here in our Punnett square. So we would see that all of our offspring are heterozygous, and if they're all heterozygous, they're all going to contain 1 copy of the dominant allele, and that dominant allele will always mask the recessive allele. Okay. So as a result, all of our offspring in the F1 generation will end up being yellow. as we see here. Okay, so we'll go ahead and cut it there. We ran out of time. And so we'll pick up here on Thursday. And then we'll kind of go through this in more detail and give a few more examples going forward.